Okay, we're going to start off talking about mutations. We're still talking about DNA, we're still talking about chromosomes, we're still into genetics, but now we're going to talk about specifically how DNA can get messed up and cause genetic diseases. Here is one example of a genetic disease. We will be talking about many more, and eventually you'll be doing a project making a pamphlet over one of the genetic disorders. And you do not have to do one that we talked about. You can pick one of your own, but I like to talk about many of them to give you an idea. This is just called Creux-de-Chat syndrome. It's due to a deletion, so something is missing, and it's a deletion on the 5th chromosome. So there's literally a gene missing. So if there's a gene missing, genes are codes for proteins, a protein's not being made. And because of the protein not being made, the larynx and glottis do not develop properly. And so when they're babies, they cry like a cat. They sound like a cat, and the Creux-de-Chat in French means cry of the cat. And they also have varied levels of mental handicaps. Okay, now we're going to look at a picture. As you can see in diagram 1, there is DNA, and we know DNA is a double helix, so it's helical shape. And then it winds around, and so you can see how from 1 to 5, the DNA is forming our chromosomes, and it's little tiny chunks on there that are our genes. So segments within the chromosome are codes for the genes, which make our proteins, which makes us who we are. Okay. Some vocabulary words. Autosome. Autosome, chromosomes, and sex chromosomes. Autosomes are the first 22 homologous chromosomes. They have nothing to do with the sex of the individual. So if you look at the karyotype off to the right, 1 through 22, our chromosomes are numbered by size. So chromosome 1 is the largest one. You get one from mom, one from dad. Number 2, you get one from mom, one from dad. Number 3, you get one from mom, one from dad, all the way to 22. Those are the autosomal chromosomes. They're forming your autosomal traits. The sex chromosomes are the 23rd pair of chromosomes that determine the sex of an individual. So in every cell, you have 2 sex chromosomes. Females are XX, males are XY. If we're talking specifically sperm, now remember the making of the sperm and the egg is the process of meiosis, and everything gets split in half. You only have half of everything. So in a sperm, you don't have 44 autosomal chromosomes and 2 sex chromosomes. You only have 22 autosomal chromosomes and 1 sex chromosome. So the male sperm is either 22X or 22Y. And then that will hook up with an egg to make the proper 44 autosomal and either XX or 44XY. In the egg, there's 22 autosomal and 1 sex chromosome. So for females, the eggs are 22X. Okay, so for a female, you get 1 X from dad and you get 1 X from mom. And if you're male, you get your X from your mom and your Y from your dad. Okay, and I discussed that last trimester. So it's the male that really determines the sex of the child, because it doesn't matter. The female is always bringing in an X. The male is going to either bring in an X or a Y, and then that determines the sex of the child. Okay, back to the autosome, body chromosomes, determining kind of traits nothing to do with sex. So genes located on the autosomes control autosomal traits and could possibly form autosomal diseases if there was mutations in these chromosomes or in these genes. And there's autosomal dominant and there's autosomal recessive, and we're going to talk about some of those later. Okay. Significance of mutations. Most are neutral. They don't really cause anything, a birthmark, something with your eye color. Some can be harmful, like all the diseases we'll be talking about. There's sickle cell anemia, Down syndrome, and some could possibly be beneficial, because sickle cell anemia, people that have that actually do not get malaria. Immunity to HIV. So there are beneficial mutations, but we really won't be talking about those. We're going to be talking about the harmful mutations and the ones causing genetic disorders. There are 2 ways in which DNA can become mutated. Mutations, you can actually inherit them from the parent to the child. So the parent had the mutation in them, they have a baby, the baby gets it. But sometimes there's parents who are normal. They don't have any mutations whatsoever. They have no disorders. So what happens in the forming of the egg or the forming of the sperm, things take place, a mutation happens, and now a child has a genetic disorder. And there's different types of mutations. There is something called a point mutation, which is a change in one or few of the nucleotides. And with point mutations, it can be a substitution, it can be an insertion, or it can be a deletion. And think about what these words mean. If you're substituting something, you're replacing it. So in this first example, we'll just say the top part is what it should read. The fat cat ate the rat. Well, now if you look, it says the fat hat ate the rat. Well, what happened is the C was replaced or substituted by the H, but you still have 1, 2, 3, 4, 5, 6 codons. Okay, here's an insertion. The fat cat ate the rat, but then now we had something inserted. The fat cat, an X got inserted in there, and now we still have ate the rat, but it's not hooked up with the proper letters it should be. Deletion. The fat cat ate the rat, but now you can see here that the letter was deleted. And so we don't see the fat cat ate the rat. Everything is moved to the left, and we would have improper codons. Insertion and deletion are also known as frame shift mutations. This would be point because one point is messed up, but with the insertion and deletion, it's also known as frame shift because if you look at this, the inserted part, the whole thing's been shifted to the right, and in the deleted part, the whole thing's been shifted to the left. So point mutation, there's one point of it got messed up. That can be substitution, insertion, or deletion. And then the insertion and deletion are also frame shift mutations. It's a shift in reading the frame of the genetic message so that proteins may not be able to perform its proper function. So again, there's an example where the H got inserted in and shifted everything to the right. Deletion, the H was deleted, everything was shifted to the left. You still have codons, but it's not the right codons. You would not be making the correct amino acids. You'd be not making the correct proteins. And I have more examples of this up on the board, which we'll be talking about in class. Okay, some other types of mutation. So I give it kind of an original, the chromosome, what it should look like. Here's a duplication. Instead of A being A, B, C, D, E, F, it's been A, B, B, C, D, E, F. And where the codons would split up, you would not be getting the right amino acids, you'd not be making the proper protein. Inversion, the word inversion means something's been inverted or flipped. So we have A, B, C, but then instead of being D, E, F, we have F, E, D. Translocation, trans means to move, we're moving location. So as you can see, we have A, B, C, but then the J, K, L, and the D, E, F switch places. So things got moved to a different location. And then there's non-disjunction, and I'll explain that in a minute, but it's how you can get a whole extra or missing chromosomes. All of these other examples, substitution, duplication, inversion, translocation, deletion, insertion, those are all just one gene getting messed up. Non-disjunction, we're talking about a whole extra or whole missing chromosome, which would be thousands of genes being messed up. So here's my little explanations for the inversion, the translocation, the duplication, and the non-disjunction. I give a much broader definition here for non-disjunction. During meiosis 1, a homologous chromosome fails to separate, or during meiosis 2, a chromatid fails to separate to produce sex cell with an extra or missing chromosome. I'll be showing you an example of this. Non-disjunction, again, when homologous chromosomes fail to separate during meiosis 1, or sister chromatids fail to separate during meiosis 2, which produces sex cells with the wrong number of chromosomes. Again, we're having whole extra, thousands of extra genes that are not needed, or thousands of missing genes. And there's many examples for this, and a lot of this has to do with plants, so we're not going to really talk about it. So I have extra information on this slide, but you really just need to understand non-disjunction. Okay, we're going to go ahead and look at the picture. So if we look at the picture, here's what it should happen. Now granted, in humans we have 23 pairs of chromosomes. Okay, we should have 46 total chromosomes, but again, it's hard to show you that. So here they're starting out with just 2 chromosomes, a homologous pair. Homologous pairs separate. The chromatids are separating, so you have 1 blue in each sex cell and 1 red, which is what should happen in meiosis. But if you look at here, at the middle section here where it says abnormal, the homologous chromosome got stuck together. So now we're making a sex cell that has 1 red and 1 blue, and that's not what it should be. And then we have some sex cells with missing chromosomes. Okay, then we get to the third one here. The first step's okay, but then when the chromatids are supposed to separate, it's not doing it correctly. So we have a blue and maybe a sperm with a blue, but then now we're missing a chromosome. And here we have 2 of the same chromosomes, and that should never happen in sex cells. You have 1 of every chromosome. So let's say we have the one with the 2 red chromosomes, that's the sperm, and it hooks up with an egg with a proper amount. You now have 3 of that chromosome, and you should only have 2 of that chromosome. And that's what the diagram up here is trying to show you again. So here, if you look at the egg, it has 3 reds, and the sperm has 2 blues, and there really should only be 2 of each. So it's coming together, and you have 2 blues. You don't have 2 reds, you have 3 red. And so you have an extra chromosome. That's non-disjunction. And there's some other lectures out there that we're going to be watching about non-disjunction. So again, mutations are change in chromosome numbers or a change in the gene itself. If we're changing the genes, we're changing the codons, we're changing the amino acid, we're changing the protein, probably not making a protein, and then things get messed up. There are the point mutation. 1 point is messed up, and that can be a substitution, insertion, or deletion. With insertions and deletions, it can cause frame shift mutations. You're shifting it to the left or to the right. There's also the duplications, where you're duplicating a base, the inversions where bases get flipped, and the translocations where bases get moved.